spearheading the validation of our technology for PROTAC screens, and he'll be the one answering all of your questions. So if you have questions, please type them in the chat, and then Kartik will answer them. He'll be very glad to answer them. I also have with me my colleague, Myra Zur. She's the sales and marketing manager, and she'll be the one following the questions and reading them to Kartik. As for my role, I'm going to be presenting this. So the title of this webinar, which we hope is going to be, we think it's going to be a regular thing for us since we've had a lot of interest. So the title of this webinar is PROTAC Drug Discovery Revolution, Bottlenecks and Solutions. And it's actually a really good title because it summarizes everything I'm going to be talking about in terms, starting by introducing the PROTAC strategy, its advantages over small molecule strategy, but also we're going to be talking about the challenges that are facing the different scientists in drug discovery, and we're going to be offering some solutions in the form of our assays and kits. So first, I'm going to start by giving a brief corporate profile for Life Sensors. We're a leading biotechnology company with a special focus on PROTAC and Ubiquitin-proteasome system. We have a catalog of around 500 products that are used worldwide by scientists in industry and academia. The products I'm going to be talking about are the PROTAC assay plates, as well as the polyubiquitin capture reagents that are known as tubes. And I also wanted to say that Life Sensors also has a CRO component through which we assist big pharmaceutical companies as well as small biotechnology startups in their drug screening endeavors through assay development. So like I mentioned, the core of Life Sensors work is related to the Ubiquitin-proteasome system, or also known as the UPS. This is a system that the cell uses for maintaining protein homeostasis. It is involved in almost in all of the cellular pathways, and it's based on an interplay between three enzymes. There is the E1 enzyme, which is responsible for activating the ubiquitin molecule. The E2 enzyme that conjugates, catalytically conjugates ubiquitin onto the target's protein via the help of the third enzyme, which is the E3 ligase. So the E3 ligase is actually considered the most important component of this enzymatic triad because it confers substrate specificity. And in most cases, the polyubiquitinated substrate gets degraded through the action of the proteasome. From a drug discovery perspective, the UPS has always been considered to be very difficult to target, and it's always also deemed undruggable for two main reasons. The fact that the E1 and the E2 enzymes are not specific, and the E3 enzyme is actually, even though it's specific, it doesn't have catalytic activity, therefore it doesn't have an active site that can be easily targeted through small molecules. So for the longest time, in terms of drug discovery, the UPS was not, considered a viable option, and that was not until the PROTAC strategy emerged. So what is the PROTAC? So PROTAC strategy is based on the use of a hetero-bifunctional molecule called PROTAC molecule. It stands for protein targeting chimera. The molecule has two ends, one end that specifically binds and recruits an E3 ligase, and the other end binds to a target protein. And the two extremities are actually bound to each other through a chemical entity called the linker. So once the PROTAC binds simultaneously to the E3 ligase and to the target protein, which is known as the ternary complex formation, by bringing these two proteins into proximity, it leads to the recruitment of a ubiquitin charged E2 that leads to the ubiquitination of the target protein. And through multiple rounds of ubiquitination, you end up with the polyubiquitination of your target protein and to its subsequent proteasomal degradation. What is also interesting about PROTAC is that once the target protein is degraded, then the PROTAC is free to bind to another E3 ligase, another molecule of the target protein, to do another cycle of polyubiquitination and degradation. And that brings us actually to the main advantage of the PROTAC molecule over traditional small molecule, is the fact that because it is recycled, then it leads to a sustained degradation, and therefore it is not needed in high doses to achieve the intended goal. And again, so that is one of the advantages. There are other advantages. For example, the fact that PROTACs only need to bind to the target protein, and they do not need an active site in the form of a pocket like small molecules do. It also doesn't need to bind to the target very strongly. It has, again, other advantages in the sense, especially when when we are trying to target proteins with multiple functions and therefore multiple domains. So it's sufficient to only bind to one domain, and then the whole protein gets degraded, and therefore you get rid of all of the functions. You can also develop a PROTAC molecule that actually can be isoform specific. And also the last advantage that we see, like it's illustrated here, is that by binding to one of the components of a complex, the PROTAC will lead to the degradation of the whole complex, not just of a subunit. So here I hope I showed that there's a lot of advantages to PROTAC mediated degradation over traditional inhibition. However, the PROTAC technology is still facing some challenges, and that is mostly due to the fact that it's obviously a new strategy. So I guess we can summarize two main challenges that are facing the PROTAC field right now. One is mostly relying on the fact that there are only four E3 ligases that have been successfully recruited through the PROTAC molecules, and actually only two of them, cereblon and VHL, that are routinely used to develop PROTACs. And so there are disadvantages linked to targeting these E3 ligases, but also the fact that even if the PROTAC is successful into bringing these E3 ligases into proximity with the target protein, then it doesn't necessarily mean that these E3 ligases are able to act on the target protein. And therefore, some targets are deemed, like I mentioned here, unprotectable. However, it does highlight the necessity to recruit novel E3 ligases for a more varied degradation of targets. But the other challenge that I think we're going to be focusing on today is more the fact that has to do more about how the PROTACs are being screened for currently. And we do feel here at Licenses that the traditional approaches are inefficient, and they are and are linked to a lot of errors. And that's why we chose to develop our own PROTAC assay. But before I get into that, I wanted to highlight the different assays that are currently used for PROTAC screens. So first you have the Western blot, which is a very unreliable technique that is time-consuming, does not provide any quantification information, and also doesn't allow you to test for multiple compounds and multiple conditions at the same time. The other technique is the reporter gene assay that is developed by Promega. It is a much better alternative to Western blot in the sense that it is high throughput. However, it does require the end user to develop stable cell lines that express the proteins of interest in a tagged form, and also at expression levels that are not reflecting of what happens or what occurs in nature. And therefore, this is a source of a lot of errors that could fail a whole drug discovery program. Again, just to reinforce the inadequacy of the Western blot technique for PROTAC drug discovery, this is actually published work where the MOLM 14 cells were treated with a PROTAC that leads to the degradation of multiple kinases at the same time. And first, we can't help but notice the fact that you can't test for multiple conditions of these different compounds at the same time. And also the fact that detecting with through a Western blot using an antibody, in most cases, it's very difficult to have a clean detection, and it makes it very difficult to identify which band corresponds to the protein of interest. And again, like I said earlier, it doesn't provide any quantitative information about the effect of these PROTACs. Another challenge actually that scientists in the PROTAC field are facing is due to the fact that PROTAC molecules do not necessarily lead to degradation. And this slide actually summarizes the different scenarios that can occur during a screen, which we also have seen in our own hands. So the first scenario on the left is the ideal scenario, where you have ternary complex formation followed by polyubiquination of the protein of the target protein, and it leads to its degradation. The other cases are due. The other scenarios that can occur are even though you have a ternary complex formation, but there is no polyubiquination, or sometimes you might have polyubiquination at the wrong lysine or with the wrong linkage, and therefore all these cases do not lead to the degradation of the target protein. So with all these challenges in mind, we wanted to develop an assay that is not only high throughput, but also can differentiate between the different events that can occur. They can also an assay that will give you information about the polyubiquination event prior to the degradation, and therefore you can, by dissecting these events, you'll be able to identify the reasons why the PROTAC is a success or a failure. So before I get into the assay, I'm going to talk about the tube technology that is actually utilized in the assay. So tubes stand for tandem ubiquitin binding entities. These are polyubiquitin capture reagents. And what they are, they're protein domains that we have engineered here at Life Sensors. And we gave them the capacity to bind and recognize polyubiquitin chains. They bind to these chains in the nanomolar range in terms of affinity. So we have commercialized pan-selective tubes, meaning they bind to all polyubiquitin chains, as well as chain-selective tubes, which bind to a certain type of polyubiquitin chain. So we have K48, K63, and linear tubes, and there's a slew of tubes targeting or binding to atypical chains that are in the pipeline, especially K6, K11, as well as K33. What is also very interesting about the tube entities is the fact that they're versatile as molecules. Therefore, you can conjugate them to different moieties. So you can conjugate them here to beads, as well as to affinity tags for purification purposes. We also conjugate them to biotin or HRP so that they could replace ubiquitin antibodies in a Western blot setting. And they can also be conjugated to fluorescent tags for imaging purposes. More importantly, we also incorporate them in our different assays, whether they're in vitro or cell-based, and that's how we developed the PROTAC assay plate. So the PROTAC assay plate is actually a high throughput assay. So it uses a microtiter plate. The wells on the plate are coated with the tube reagents, which are polyubiquitin capture reagents. And when you apply your cell lysate, for example, after treating with a PROTAC, all of the polyubiquinated proteins are trapped on the plate. And then you can assess for the polyubiquination level of your protein of interest using a specific antibody that is conjugated to a signal, and then you can follow that signal using a plate reader. So we started, or actually Kartik here started, developed a plan of experiments to validate this assay for PROTAC screen. And the first experiment he did was treating Jurkat cells with DBET1 PROTAC. So this is a PROTAC that is very well established. It recruits cereblon as an E3 ligase and leads to the degradation, polyubiquination and degradation of bromodomain proteins, including BRD4 and BRD3. Here we have we analyzed the cell lysis generated from this treatment by Western blot. So we have a dose response experiment on the left, as well as a kinetic experiment on the left. And in both cases, we see that there is a decrease in protein levels of BRD3 in this case, in a dose-dependent manner and also over time. Again, here I chose to use the word or the expression protein levels because the Western blot is limited in the information it provides. We don't know if this decrease in protein levels or the band corresponding to BRD3 is due to degradation or to the fact that there is polyubiquination, but the antibody, like in most cases, is not sensitive enough to detect for the latter that corresponds to polyubiquinated proteins. So analyzing the same samples using our PROTAC plate assay, we can see that in the dose response experiment shown on the left, that there is a peak around 1 micromolar corresponding to maximum polyubiquination, and then a decrease in signal when we increase the PROTAC concentration. And that is due because at a fixed time, once you when you increase the amount of PROTAC, then you miss the polyubiquination window and you're in the degradation phase. In the kinetic experiment shown on the right, the condition corresponding to PROTAC treatment only is shown through the blue line. And we can see that over time, we see that there is a maximum polyubiquination peak that is followed by disappearance of signal, which corresponds to proteasomal degradation. And in order to confirm that, we co-treated with MG132, which is the proteasome inhibitor, shown with the red lines, and we can see that at every time point there is accumulation of polyubiquinated form of BRD3, thus confirming that the disappearance of signal is actually happening because of proteasomal degradation. So with this first experiment, we established that we had a PROTAC assay plate that not only is high throughput, but also provides a link between the polyubiquination process and the degradation process. To further validate this this assay, we treated K562 cells with another established PROTAC. It's called the multikinase PROTAC. This is a PROTAC that recruits cereblon again as an E3 ligase for the polyubiquination or the degradation of a significant number or numerous kinases at the same time. Two kinases that we chose to follow are BT kinase and Aurora kinase, and we can see that in our hands it does lead to a decrease in the protein levels of these kinases over time when we treat K562 cells with the PROTAC molecule. Again, more interestingly, using analyzing the same cell lysate and through our own technology, we can see that we can follow the polyubiquination and degradation of the both targets. And in addition to that, we're able to see that Aurora kinase seems to be a more sensitive target of PROTAC compared to BTK, as the degradation happens before then that of BTK kinase. And so with this experiment, we're also very happy to say that not only we had an assay that provides more information than the traditional methods, but we're also happy to say that this assay is compatible with multiplex detection. And one of the final steps to, again, validate this assay for PROTAC screen, we wanted to verify that the results that we have seen using the multikinase PROTAC are reproducible by an alternative way of detection. And that's why we resorted to using the tube-based proteomics. So this is a service and also a platform that combines tube enrichment with traditional mass spectrometry. So when we get our the samples, which are frozen pellets of frozen tissue, we perform the cell lysis, and then we incubate with the tubes. That would allow us to enrich for polyubiquinated proteins. The latter get eluted and subjected to tryptic digest, and then we submit all the samples for mass spectrometric analysis. So this is the result of that experiment. The red dots correspond to the PROTAC only condition, and we can see that there's a significant number of kinases for which the polyubiquitination level is reduced in response to PROTAC treatment. And to confirm that this decrease in polyubiquitination is due to degradation, we co-treated with MG132. Again, it's a proteasome inhibitor, and that is shown by, well, represented by the black squares. And we can see that, again, a significant number of those kinases are actually rescued by proteasomal degradation. And then out of those kinases, we're happy to see that that list includes aura kinase as well as BT kinase. And in the same fashion that we saw with our plate-based assay, we see that aura kinase is a very sensitive target of this PROTAC, whereas BTK is more of a late or more resistant target of that PROTAC. So here at Licensors, we pride ourselves for being very flexible to our clients' needs, and a lot of our clients are biotechnology startups, and they do not have a biology team, and therefore they're not equipped for cell culture. So for that, we have developed an in vitro PROTAC screening platform, which is, again, a high throughput assay that functions with the same principle as the cell-based one, where the wells in the plate are coated with the tube capture reagent, or sorry, the tube, which is a polyubiquitin capture reagent. And then instead of applying a cell lysate, then you perform an in vitro polyubiquitination reaction. So basically, you would need purified form of E1, E2, ubiquitin, E3 ligase, as well as a pure form of your substrate, and you would include your PROTAC molecules. So as the substrate gets polyubiquitinated, it gets trapped on the plate, and then you will assess for the polyubiquitination signal or the polyubiquitination level using a specific antibody that is conjugated to a signal. And a way to show that also you have polyubiquitination, but you also have ternary complex formation, you can perform a dose-response experiment where by adding increased amount of PROTAC, you have this a bell-shaped curve where there is a maximum polyubiquitination signal corresponding to a maximum ternary complex formation. But then as you increase the amount of PROTAC, the ternary complex gets disrupted, and then you would have binary complexes, and then there's a loss of polyubiquitination signal, which is known in the field as the hook effect. So I will show you one experiment that we use to validate this in vitro assay. For this experiment, we use two PROTAC molecules, DBET1 and DBET6. Both of them recruit cereblon and lead to the degradation of bromodomain proteins, like we see here by Western blot. And then so basically in vitro, they would lead to the polyubiquitination of BRD4, which is the target that we chose here. And the only difference between these two molecules is the length of the linker. DBET6 has a longer linker, and it was shown in literature to be a more potent degrader of BRD4 than DBET1. So in both cases, we get to see that there is, as we increase the amount of the respective PROTACs, we see that there is a maximum polyubiquitination corresponding to ideal conditions of ternary complex formation. And then as we increase the amount of PROTACs, there is disappearance of the polyubiquitination signal, which is the hook effect. And as was shown in the literature, DBET6 is more potent than DBET1 for the polyubiquitination of BRD4. So this is the end of the part about our PROTAC assay. Since like I told you, we have seen a lot of interest in our webinars. I just wanted to give you a small preview about one of our product launch, for which we're going to have a webinar hopefully by late fall or beginning of the winter season. And then so this is to streamline, as we finish streamlining the detection part of a screen, we also wanted to streamline and make the sample preparation part more efficient. And for that, we have developed a high throughput sample prep plate, which in the wells, the wells have filters, and in the filters you have our tube reagents. So basically, it will allow you to do the tube pull down and elute your polyubiquitinated proteins for 96 conditions at the same time. And once you get the elution fraction, which is the polyubiquitinated proteins, we have done experiments where we showed that they are compatible with Western blot analysis, even though we wanted to stay away from the Western blot, as well as mass spectrometric analysis and any activity assay that you choose to perform following the elution. So with that, I just wanted to summarize this webinar, and hopefully I'll get you to share our excitement about the PROTAC field, because it is a very smart strategy that gives a lot of advantages over traditional small molecule targeting of proteins. And we also wanted to give you the challenges that are facing or that are part of this field. And we firmly believe that our platform, whether it's in vitro or in vivo, is going to be able to speed up any PROTAC drug discovery screen. And with that, we are happy to answer any of your questions. If you have any questions after this webinar, do not hesitate to reach out to either our scientists or our business development team, and we'll be happy to answer any questions or jump on a call with you guys to see if we can help you with any services or any products, or even just give you advice on how to plan your experiments.
Okay, so we do have some questions. Dimitri did have a question, but he said it was later noted in the webinar, so that was wonderful. Next up, we have Saheen. So one of the questions he did ask was, how is the cell permeability of PROTACs in general? So that's the first question.
Okay. Yeah, so I can start with giving an answer, and then Kartik can add. So basically, that is a really good question, because it is kind of part of the history of PROTAC. So when PROTAC actually started, it was a peptide based, where the recruitment of the E3 ligase, which was VHL at the time, was used through a peptide. And that was actually the first PROTAC that was, not discovered or created or designed. However, they did, once they went into in vivo, they saw that the permeability was very low, because it was peptide based, and that's how PROTAC transformed from being peptide based to small molecule based. I think Kartik can say more about how it compares to a regular small molecule.
Yeah, definitely. So compared to small molecules, since PROTAC has two warheads and then a linker, it's slightly bigger than a small molecule. So there will be some kind of permeability issue, but a lot of scientists are trying to figure out a better seal of P for these molecules to get better permeability. If you see our examples from our in vitro assay, where you see DBET1 and DBET6, that is one of the issues why you see better degradation for DBET6. DBET6 with a longer carbon chain length is about 100-fold better in terms of cell permeability compared to DBET1. That rightly translates to about 10 to 50-fold increase in PROTAC degradation efficiency for DBET6. So definitely permeability is a big concern for the chemists to figure out what is the best best configuration to have, what is the chain linker length, what are the small molecules that you're using as a warhead that will allow you to get into cell much better, to have much better degradation profiles. So yeah, you got to have a idea about how you're going to handle the issue as the small or the PROTAC becomes larger with adding molecules into the chemistry design.
Yeah. Okay. And the next question that he did ask is, if there was a prediction tool to find out the E3 ubiquitin ligase is leading to the degradation of the protein of interest.
Is it a prediction tool to find it?
Yeah, I think that is one of the things that is lacking. Again, why I guess PROTAC molecules are slow to roll in the clinic is the fact that because it's a new strategy, that there are no rules actually that we know about first of all, how the PROTAC should look like, what the linker should look like, and which E3 ligase is going to be more potent into degrading it or leading to its degradation. I think a lot of it can be, and of course, if you think of that, if it's probably a lot of crystal structure that could predict that the PROTAC will lead your protein to be in the right orientation.
Exactly, yeah.
That would lead the E2 to come and because it's important to be polyubiquitinated at the right lysine and with the right linkage. Yeah.
Yeah. One easy answer is you have to make sure you're oriented right, and there is a chance of ubiquitination the way that you're designing your small molecule. It's more or less have to be a little bit flexible so that you will be able to ubiquitinate your target. But it's more chemistry driven. So what we could say is at this point there are no set tools to really predict this, but there's a lot of work happening in the virtual companies where they're trying to screen the large libraries and do that kind of stuff.
Yeah.
So.
Yeah, and I guess I just wanted to point out that the fact that there are no rules, so I guess scientists or companies will have to screen with a lot of compounds. And then see which one will give you a hit. And that's what makes having an efficient and the correct screening tool more important, because you don't want to be using a Western blot if you have a library of PROTACs to screen for, because you can't predict which one is likely to lead to the intended effect.
It'll be very labor intensive.
Yeah.
And from Jay Min, had reached out asking if we had a chance to compare our ubiquitome with global proteomics results.
Not in the particular experiment we have shown, because we took the traditional PROTAC, and there are a lot of studies out there that did the total ubiquitome that showed selective degradation of these bromodomain proteins, and as well as the kinase. So we have a reference in the webinar. You can go there. There is a global ubiquitome profile that was performed by the Dr. Nathaniel Gray's group by themselves. So you could follow that, and you can obviously compare with our ubiquitome that we have. We're very happy to assist you guys to do that comparison if you're interested. But there's a lot of studies out there that shows the total kinase degradation profiles from total proteome versus us doing the ubiquitome.
Mm-hmm.
Yeah. And he asked some more questions. He asked if our plate only provides a 96 well format, as we don't ask if people have a 384 format.
Yeah, so this assay has been validated for 384 format as well. I'm not sure if on our website we provide the 384 well format.
I just want to make sure. So are we talking? So let's talk first about the cell-based, the PROTAC assay plate, not the in vitro.
That's what he's asking.
Okay, all right. So
it's UV plate, yes.
Okay. So that one, it has been validated for 384.
Yes.
Okay, because we only make it available as 96.
96, yes.
Yeah.
Because majority of our clients choose to use, but we can definitely offer.
Okay, so we can adapt it. It's not a custom.
No, yes.
Okay, so we can definitely adapt it to a 384 format.
Yeah, just to reach out to us if that's something that you were interested in. Reach out to our BD team or our customer service, and we can definitely format to customize it to your needs. Within that, he did ask a very good question: what's the advantage of our in vitro PROTAC screening over alpha screen?
Yeah, so the most important advantage is following the ubiquitination. As far as I see, like if you're mentioning alpha screen, you're trying to talk about target recruitment in terms of PROTAC between the E3 ligase and target, right? So that wouldn't give you information about what is the rate of ubiquitination, what is the level of ubiquitination in terms of your PROTAC. So our in vitro assay, while establishing the ternary complex, it also establishes, whether your PROTAC allows your target protein and the E3 ligase to get in right orientation to see the ubiquitination profiles. So that'll give you much more information because not all, if you see the first few slides, you'll see the challenges with the PROTAC. Even though there will be recruitment of E3 ligase and your substrate, not all of that recruitment leads to ubiquitination subsequent degradation. So you'll be benefited from getting that extra information that whether you'll be able to do the ubiquitination or not.
I think there's also a question we missed from them. Did you encounter any technical issue using the 384 well format for the ubiquitin plate screen?
No, not that. I mean, the technical issue would be like, how do you choose your controls to make sure what you get is a reliable, reproducible signal. So that is one thing that you have to design by yourself, what kind of controls you're planning to use within your plate to make sure the data that you're generating is reliable for your screen that you're trying to achieve.
Great. Awesome. Thank you guys so much for all of your questions.
Do you have any other questions?
Yeah, and we'd love to have some more if you guys would like some more. We'll give you guys a few more minutes to type them out. If not, I am very happy to announce that we did choose our winner of the Amazon gift card. We did pick out a random name for those who have RSVP'd, so I do hope they're on here right now. It is to a Mr. Stephen White. We will be reaching out to you shortly with your email that you provided using the RSVP, and we'll be sending it over. So thank you so much. Again, we'll stay on here for an extra minute or so and see if you have more questions. If you have any more questions that you feel like you have for later on, if you think of anything else later on, please, again, feel free to reach out to bd@lifesciences.com or customer service at lifesciences.com, and we will be happy to address any questions that you have regarding our PROTAC assay plate.
Thank you.
All right. Well, thank you so much, guys.
Thank
have a
you.
lovely day.
Bye-bye.
Thank you.
Thank you, guys.
Bye.
Bye.